there's anything that needs to be shown that's getting cut off, I'll just make it not full screen. Okay?
Okay.
Right. So
Can you go to the next slide? Okay, so apoptosis, which is known as programmed cell death and even as cellular suicide, is a process that allows cells that aren't behaving as they should to self-destruct. Otherwise, these faulty and misbehaving cells can potentially cause problems to an organism and its survival if it were to be kept inside instead of being destroyed. This can sometimes be confused with necrosis, which is another form of cell death. However, in this case, cells die due to injury and it can also result in inflammation. These cells are killed by something harmful rather than being triggered by some sort of signal that tells them that they need to undergo cell death. Apoptosis is an extremely important and crucial process, and organisms would not be able to function normally without this sort of mechanism. Apoptosis is used for both development and defense. And as for development, it occurs naturally as organisms develop. One example, you can look at the bottom diagram. The mouse paw initially starts off in more of a rounded shape, but apoptosis occurs during development, and that allows for the formation of these intricate shape and features of the paw. So cells in certain areas self-destruct, which provides the resulting unique shape. In this case, cells are destroyed not because they're causing harm, but because they just need to be deleted during development in order to form the proper structure of the organism. It is also used as a sort of defense system where a faulty cell can undergo apoptosis if it is misbehaving and potentially serving as a threat to the organism. And in turn, this will benefit the organism by keeping it safe. Cells that may need to undergo apoptosis include cells that may be abnormal or cells with DNA damage, and their presence may hurt the rest of the organism, which is why they may need to be destroyed. Apart from development and defense, apoptosis is also important to other cellular processes, including cell turnover, the development of the immune system, and embryonic development. Next slide. So you can go to the next slide, actually. So you guys can look at a visual representation of how it works while I discuss basically an overview of it. So there must be a signal that triggers a cell to let it know that it needs to undergo apoptosis. And there's a variety of stimuli that can trigger it, and this can include physiological or pathological signals as well as external or internal signals. This process can actually be initiated by two distinct pathways, the extrinsic or intrinsic, which will be discussed in detail later in the presentation. So then cellular agents are, they fragment all material inside the cell and then they chop it up into tiny pieces and chunks, and then the cell will also shrink and form bubble-like protrusions, which are known as blebs. And then the fragmented pieces will pinch off in membrane-bound apoptotic bodies that will release signals that attract phagocytic immune cells or scavenger cells to digest it. Next slide. Although apoptosis is the death of a cell, it does not mean that it's out of control or if it's a chaotic process. Besides being a critical mechanism, it is also a normal and controlled part of the development of an organism, as I've mentioned before. Some examples of this include the removal of cells in an orderly and controlled manner. So this refers back to the previous example on how certain cells can be deleted to form intricate features like the fingers we have today. And it also eliminates potentially threatening cells. Whether a cell has a mutation or some sort of damage that causes it to be harmful, apoptosis can be triggered to destroy these cells. And overall, this process helps to maintain homeostasis by serving as a defense mechanism that helps keep safety in a cell and by being a regular part of development. And what I just want to emphasize is that the process of apoptosis is really important for a cell to have the ability to self-destruct if it ever acts as a potential threat to the overall survival of an organism. Next.
okay, so what would happen if apoptosis couldn't be carried out properly? in the absence of proper apoptosis, it can threaten components that may be incorporated into other cells. Like if it's not uptaken correctly, then other cells can consume what is supposed to be a dead cell, and then that would disrupt the healthy cell. And that is how mutations can arise and threaten good cells. And then if enzymes can't carry out apoptosis, then it can basically cause cancer. Some complications would be neurodegenerative disease, which is Alzheimer's, and then autoimmune disorders and cancers. Basically, how this happens is like associated with caspases, and it suggests that inappropriate activation or like dysregulation of cell death and inflammatory pathways that control these things like get messed up, and then can cause apoptosis in cells that shouldn't be dying or basically going through the apoptosis process. And then how this happens starts with, it's called the caspase. It's a family of cysteine proteases, and these enzymes basically degrade proteins at specific aspartic acids. Inactive caspases are known as procaspases, and then they're activated by other caspases. This results in a proteolytic cascade. And then in the diagrams, you can see that this is the inactive one, the inactive procaspase, and then it's activated by the cleavage. This breaks off and creates an active caspase. And then this just continues to happen to create what is known as the caspase cascade.
So the caspase cascade is primarily an event where caspases respond in succession when activated by an apoptotic stimuli, and this leads to proteolysis, cell assembly, and apoptosis at the very end, so cell death. Yeah. So this can be triggered by stimuli from the cell surface or from the mitochondria. The image shown below it shows an example of the Faz death receptor, but it can be triggered by other things such as the TNF, GRB, DNA damage, and a few others. And these are all linked with initiator caspases. And when these caspases are stimulated, the initiator caspases further cleave and trigger downstream executioner caspases, which then cleave other proteins, and these events eventually result in cell death. The caspase cascade pathway plays a key role in several neurodegenerative disorders like we talked about earlier, like Parkinson's and Alzheimer's, but also tumorigenesis and autoimmune disorders. Next slide.
So there's, as mentioned before, there's extrinsic and intrinsic. So the extrinsic pathway that initiates apoptosis is triggered by a death ligand binding to a death receptor, such as TNF alpha to TNFR1. And this was a lot to fit in the slides, but you could kind of see the outline in the slides. The TNFR family is basically a large family that consists of 29 transmembrane receptor proteins. And they're basically organized in homotrimers and activated by binding of the respective ligands. They share similar cysteine-rich extracellular domains and have a cytoplasmic domain of about 80 amino acids called the death domain. This death domain plays a critical role in transmitting the death signal from the cell surface to the intracellular signaling pathways. And there are about 19 members of the TNF ligand family, and binding may result in a number of responses, including proliferation, inflammation, and apoptosis. And this depends on the adaptor proteins associated with the activated receptor. TNFR may also stimulate pro-inflammatory pathways, and that leads to activation of NFKB, and that happens because of recruitment of the RIP. And other than TNFR1, the FAS and DR4/DR5 is also involved in the pathway, and they're involved as death receptors, and they bind to CD95 and TRAIL trail, respectively. And all of these ligand binding to receptors will lead with the help of adapter proteins to recruitment, dimerization, and activation of a caspase cascade that was just mentioned. And this eventually leads to the cleavage of both cytoplasmic and nuclear substrates. So why is this important? Apoptosis as a whole is a process that basically where cells undergo programmed death and is a counterbalance to proliferation. This prevents inflammation of the surrounding tissue, basically, and apoptosis relies on activation of distinct signaling pathways that are often deregulated in cancer. So this plays a huge role in cancer, and basically it offers a unique therapeutic strategy to induce apoptosis in cancer cells, which is what you need. Hence, our ability to exploit these pathways to design more effective and non-toxic therapies for cancer is dependent on our understanding of the biological mechanisms. Next. So now that we've covered extrinsic, there is also the intrinsic apoptosis pathway. So the intrinsic apoptosis pathway is initiated, by example, by chemotherapy and or radiotherapy. It is activated by a range of exogenous and endogenous stimuli, and that could be things such as DNA damage, oxidative stress, and it plays an important function in development and in the elimination of damaged cells. In the intrinsic pathway, the functional consequence of pro-apoptotic signaling is mitochondrial membrane perturbation and release of cytochrome C in the cytoplasm. And this is where it forms a complex or a apoptosome with apoptotic protease activating factor. And the intrinsic pathway basically induces apoptosis by directly activating caspase-3 or by cleaving VID, which results in mitochondrial dysfunction and subsequent release of cytochrome C and activation of caspases 9 and caspases 3. So most chemotherapeutic and targeted cancer therapies, they kill tumoric cells through the generation of pro-death signaling, and this is what initiates the intrinsic apoptotic pathway of programmed cell death. And then I think I mentioned, how it activates caspase-3 and by cleaving VID. And again, this is another important factor in cancer and in pathophysiology, which I just mentioned with chemotherapeutic and targeted cancer. Next. So BCL plays an important role in apoptosis. The BCL2 protein family basically determines whether a cell commits to apoptosis and hence has crucial roles in development, tissue homeostasis, and immunity. The alteration of this control can either promote cancer and autoimmune diseases or augment other conditions and contribute to degenerative disorders, so basically too much apoptosis. And so in other words, it basically inhibits apoptosis by increasing the time to death and intrinsic cell-to-cell variations in the mitochondrial pathway of the cell death. Another factor also, the life or death decision for a cell is mainly determined by the interactions between the 3 fractions of the BCL2 family, and they're basically the subfamily. Examples would be BCL2, BCLXL, and MCL1, and the other 2 pro-apoptotic factions. And they basically convey various cytotoxic signals and the death effectors BAX and BAK. And these can convert into homo-oligomers, that perforate the mitochondrial outer membrane, and this triggers the proteolytic cascade that demolishes the cell. And basically, there have been insights that have shown how BAX and BAK convert from interglobular monomers into lipo-oligomers, and that basically permeabilizes the mitochondrial outer membrane. So at the end of the day, like the BCL2 family basically acts as a critical life to death decision point, and it's a very common point in the pathway of apoptosis. Um, Next.
So there are a few ways that apoptosis can be inhibited. So the first way, if it wasn't obvious enough, is called inhibitors of apoptosis, or IAPs. Um, so capsaces, which normally induce apoptosis, can be, or procapsaces can spontaneously be cleaved of their prodomains, thereby activating them. If they were to keep going uninhibited, they would have um, triggered apoptosis, but IAPs exist in the cytosol to basically prevent spontaneously activated procapsaces from going on and initiating apoptosis. And so when there is an apoptotic stimulus, to keep these IAPs from inhibiting capsaces from doing their jobs, there are also anti-IAPs that are produced and released into the cytosol to bind to these IAPs so that they can't bind to capsaces, and then apoptosis can then occur. Next slide. The second way that apoptosis can be inhibited is by these extracellular survival factors. There are three ways, the first of which is an increase in production of anti-apoptotic BCL2 proteins. So what happens is usually a survival factor will bind and activate to a receptor on this lipid bilayer, and then this will further activate a gene regulatory protein, which then then migrates into the nucleus and binds to its respective gene. This gene is then transcribed and then translated into a BCL2 protein, which then blocks apoptosis. The next is the inactivation of pro-apoptotic proteins. In this case, the survival factor again binds to a receptor, which then activates an AKT kinase, and this kinase's job is to basically inactivate this bad protein. What happens when when a bad protein is active? It'll inactivate the BCL2 protein, which blocks apoptosis. But since since the bad protein itself is blocked or inactive by that phosphate, um, the active BCL2 protein can then go ahead and block apoptosis, just as in the first one. And then finally, the third method is an inactivation of anti-IAPs. So in this case, the activated receptor will basically increase activity with the MAP kinase, which then inhibits this HID protein. And the purpose of this HID protein is to inactivate IAPs. But since the HID itself is become inactivated, the IAPs can go ahead and block apoptosis. And that should be it. Thank you guys for listening.
That's everything. Thank you guys. Yes.
Apoptosis is really important for the development of an organism, like we talked in the first example. with the diagram, during development, apoptosis actually occurs naturally to delete certain cells. Like in this diagram, in the little spaces in between, they're being deleted to form the proper shape of what the organism is supposed to have in the structure. And that also occurs in other organisms too. Like with us, our cells are also deleted in between our fingers so that we have these intricate structures.
Yes. One of our questions was, is the failure to carry out proper apoptosis, is it caused by random mutations in the cell, or can it be triggered by certain signal molecules or pathways?
It can be caused by a variety of things, but mutations or some type of DNA damage could be one of the causes. In that case, if apoptosis can't properly be carried out, then that would result in even more damage.
Or if the cell was sorry about that, guys. If the soul was carrying out apoptosis and like there was a disruption, like that can contribute to tumor metastasis and like the development of autoimmune diseases. And then the acceleration of apoptosis can cause neurodegenerative diseases. So like if it just basically isn't carried out properly in a timely manner, then things can go wrong.
Okay, thank you.
Hello? Can you guys hear me? Can you hear me?
Yes.
Oh, okay. So if apoptosis is important for development and also to stop cancer, is there any other processes that it's important for?
I mean, overall, like it can stop cancer, but that, how do you say, like cancer, it's supposed to just get rid of cells that either don't work anymore or, you know, that are dead. Like, and if it doesn't get rid of, if the process doesn't happen, then cancer can occur, right? Because what is cancer?
Okay.
And yeah, did that answer your question?
Yeah, thank you.
We wanted to know if you guys could further explain how apoptosis plays a role in Alzheimer's.
So yeah, basically, like, it could be like inappropriate activation of caspases, which is what we talked about, like this happening. Can you see the slide? So like if some cells are activated that aren't supposed to be activated, or active, basically stimulated to go through apoptosis when they shouldn't be going through apoptosis, they shouldn't be dying off, but they do, that is what causes like diseases like Alzheimer's. Does that make sense?
Yeah, thank you.
For BCL2, I just know like it's basically like the main function, or it basically determines whether a cell commits to apoptosis. Oh, actually,
Is it? It's B cell lymphoma.
yeah, or the full name, B cell lymphoma 2. like why it's named that. Because like lymphoma, like cancer. Mm-hmm.
Yes.
Yeah.
Mariah. Veronica is actually she dropped out of the class like a while back, so it's just us three. Yeah. Okay. Okay. Can you guys see my screen?
Yeah.
Yeah. Okay. So are we all ready, Carlos and
Yeah.
Chen? Okay. Right, so okay, hi guys, my name is Mariah, and I will be doing the introductory part of this presentation, which is on genetics and biology of cancer. So before I start, I kind of wanted to give you guys like an outline of how we kind of broke up our presentation. So I'll be just doing kind of like a brief overview of the development and progression of cancer. And Carlos and Chen will be going into the molecular mechanisms of cancer. So they'll be discussing proto-oncogenes and tumor suppressors and how they link to cancer. So first off, what is cancer? So cancer is an uncontrolled proliferation of a cell that migrates and invades other tissues. So this diagram over here, you can see that these cancer cells over here have proliferated out of control, and this in turn gives rise to a tumor. And you have a benign tumor and another tumor called malignant tumor. And the difference between these tumors is that cancer cells in the benign tumor are localized. So you see they haven't migrated out of the tissue. When the malignant tumor, they were able to communicate with these cells and migrate and invade surrounding tissues. Okay, so here are just some causes of cancer. So if you were to take a closer look into a cancer cell, you would notice that a lot of the time somatic mutations have occurred in the cancer cells. So chromosome abnormalities are the most common one. In class, the professor gave an example of a Philadelphia chromosome, which is this one right here. This happened because of a translocation between these two arms that resulted in a segment that encodes for proteins that lead to leukemia, which is a type of blood cancer. And then you have some other chromosome abnormalities such as deletions, duplications, and inversions, point mutations, and so on. Some outside factors are like carcinogens, ionizing radiation, so like X-rays or UV rays, and viral DNA insertions. And these are just a few to name. There are a lot more causes of cancer than what I've listed up here. And so basically, these somatic mutations kind of kickstart the ability for a cell to become cancerous because they can lead to inactivation of a gene or overexpression of a gene. And you will see this in the next slide, how this plays a role in cancer development. And so now this is a little summary right here on how cancer develops and progresses. First off, I did this in a stepwise manner, but some of these are work simultaneously. They go hand in hand with each other. So first you have a healthy cell. This healthy cell undergoes a somatic mutation. So like what I discussed above, it could be like a chromosomal abnormality or an outside factor like carcinogens. And now these mutations, so step two, so these mutations result in a genetic or epigenetic change in the DNA or chromatin, which in turn, depending on the cancer, it can overexpress or inactivate a gene, such as the tumor suppressor genes. And this now, with the combination of overexpression or activation of a cancer gene along with other proteins, allows it to bypass certain checkpoints, and the cell can continue to proliferate because it's unable to exit outside of the cell cycle. And as the cell proliferates, so in this diagram right here, you can see that as the cell proliferates, it accumulates more mutations. So this cell has one mutation, it starts to accumulate a second mutation, a third mutation, and this is advantageous to the cell because each time that it accumulates more mutations, it's able to divide faster. So at one point, this cell has accumulated many mutations that allows it to acquire an advantageous trait to where it's able to actually breach the basal lamina and go ahead and spread into other tissues. And this leads to metastasis. And with metastasis, what happens is you have a cell proliferation that occurs in this diagram right here down below, where my arrow is at. And this cell proliferation has turned malignant because now it's able to cross the basal lamina and enter into the blood, where it's able to connect to a cell wall and communicate with the surrounding cells, maybe perhaps through the CAM molecules that we learned or through the ECM components, and then kind of intercalate between the cells. And now this is its new residing place where the cancer cells can now proliferate more and add to the spread of cancer. So this process is known as metastasis. And so I just did kind of like a big picture of the development and progression of cancer. Now Carlos and Chen will go over what really happens in this area right here in step 3. They'll be talking about the inactivation or overexpression of a gene, so the oncogenes and the tumor suppressors.
All right, so there's two categories of genes that are critical to cancer, and they are oncogenes and tumor suppressor genes. And so oncogenes, what they do is they send out proliferation signals, which are signals that tell the cell to divide and grow. And they act as petals because they continuously tell the cell to divide, and which could lead to an abnormal cell proliferation. And the way they communicate with the cell is through they take part in the cell transduction pathway. Now, tumor suppressor genes, they send out arrest signals, and they act as brakes because they stop the cell cycle once they detect a mutation within a gene. So therefore, they take part in the cell cycle control and DNA repair. Now, overactivity of oncogenes and underactivity of suppressor genes That would lead to abnormal cell proliferation because the oncogenes will keep telling the cell to divide, and if the tumor suppressor genes are not active, they're not going to tell the cell cycle to stop so they could repair the DNA. And so that eventually just leads to more and more cells, which becomes an abnormal cell proliferation. Next. All right, so to go into more depth on oncogenes, so oncogenes, they originally derived from proto-oncogenes. Proto-oncogenes, they are normal genes that provide signals for cell division. And the way proto-oncogenes become oncogenes is that they go through a gain-of-function mutation. And this gain-of-function mutation makes the protein much more active. And so, yeah, that's why you see in the little image, there's like a car where once the gene is mutated, the um, the oncogene, they're known as petals because it just keeps going. It's always on, and it just keeps dividing. The cell keeps dividing and growing. So yeah, and then so proto-oncogenes, they function in cell signaling pathways, but once they are converted into oncogenes, they are activated. The pathway is activated in an unregulated way. In the image, you can see how the first cell, it's in rest. It's a normal resting cell. There's nothing active, nothing's going on. But then once a growth factor comes in, the receptor activates first, then the intracellular signaling proteins, and that would lead to the transcription of cells to divide, or cell division genes, my bad. And so that would be the normal proliferation of a cell. And then in a mutated one, an oncogene, what would happen is there would be no growth factor, but the intracellular signaling proteins, they would always be active. They would be continuously active, like they're stuck in a hyperactive state, and that causes them to continuously send signals to genes to the gene regulatory protein, and that leads to more transcriptions of cell division genes, and that eventually it becomes an abnormal cell proliferation, and that leads to cancer. Next. And so yeah, so how do proto-oncogenes become oncogenes? Well, through what process? So the way oncogenes, they result from mutations that increase the expression level or activity of a proto-oncogene. So as I mentioned earlier, oh sorry, my bad. No, so the ways they get activated is through point mutation. Mariah mentioned that earlier, how there's different mutations within the chromosome, DNA amplification, chromosome rearrangement, and other epigenetic ways. In point mutation, deletion, or insertion, that leads to hyperactive gene products. And once that's active, it's mutated, it can't be turned off. And gene amplification, that leads to chromosomal copies of a proto-oncogene. And chromosomal rearrangement, that leads to a fusion between a proto-oncogene and a second gene, which produces a protein with oncogenic activity. And an example would be I believe it was called the Philadelphia chromosome, where the one end of the chromosome 9 exchanged with one end of the chromosome 22. And when the chromosome ends fuse, so do the genes, and that leads to a oncogenetic protein. Next. All right, so an example of an oncogene would be the RAS protein. And the RAS protein is a guanine anucleotide binding protein, or G protein, or GTPase. And the cycle, it has two cycles, the conformational states. And in its an active state, it binds to GDP. In its active state, it binds to GTP. And it's involved in cell proliferation and differentiation. So then what happens when the RAS protein is mutated? It blocks GTP hydrolysis. And in the image here in the bottom, you can see the process where the signaling molecule will activate the receptor tyrosine kinase. Originally, this is how the RAS protein works. That would lead to an adapter protein binding to the phosphates on the receptor, and the adapter protein binds to a RAS activating protein, and that would activate the RAS GDP bound protein to become RAS GTP bound, and that activates the RAF, leading to a MAP kinase cascade, activating MYC, or the MYC, and also activating cyclin D, which tells the cell to divide, keep on dividing. So when the RAS becomes mutated and becomes an oncogene, what happens is originally, forgot to mention, once this whole process is done, the RAS protein would hydrolyze itself and become GDP again. But once it's mutated as an oncogene, the RAS bound GTP bound RAS protein, sorry, it would not hydrolyze itself and it will remain active. And if it remains active, that means the RAF is always active, the kinase cascade is active, MYC and cyclin D, and that would just lead to the cell to divide even more, it keeps growing, and that just eventually leads to abnormal cell proliferation, and that leads to cancer.
Next, we want to cover the tumor suppressor gene, also known as anti-oncogene. As the name indicated, tumor suppressor gene are genes that are critical in controlling and suppressing the formation and development of tumors. They are part of the cell cycle checkpoints and DNA repair system. Examples include the cyclin-dependent kinase inhibitors and RB gene, as well as p53 gene. They act as a brake, which can cause cell arrest, and underactivity of this gene can lead to abnormal this cell's gene is dominant, which means they only need one copy to be functional. As you can see on the top right diagram, when there are one mutation of this gene, the brake is still functional, still able to stop the car. But if there are two mutations of this tumor suppressor gene, the car fails to stop and can lead to like the development of tumor and cancer. And there are multiple ways to eliminate or inactivate those tumor suppressor gene, including the point mutation of this gene, deletion, gene conversion, as well as non-disjunction of chromosome during meiosis lead to the loss of the whole chromosome. And there are also other epigenetic ways that can use to control the expression of this gene. Next slide, please. So epigenetic, unlike genomic alterations, which is the direct alteration of DNA sequence, epigenetic alteration, they does not alter the DNA sequence, but they alter the way the genetic code are expressed by modifying the structure of chromosome, as well as modifying the DNA, histone, and other protein that participate in transcriptions. One of the examples shown here is DNA methylations. It shows how the methylation of DNA sequence can lead to the silencing of transcription of tumor suppressor gene. As is shown here, there are two types of cells, normal cells and cancer cells. In normal cell, the promoter region is unmethylated, which leads to the activation or transcription of tumor suppressor gene. However, in cancer cells, the promoter region is highly methylated, which in turn silencing the transcription of tumor suppressor gene and lead to the development of tumor and cancer. And to give you a sense of how hypermethylation related to the tumor formation, you can see on the bottom right diagram, it shows the frequency of hypermethylations of different types of tumor suppressor gene in different types of tumors. And you can see even in one type of tumor, they can have multiple tumor suppressor gene are hypermethylated in the same time. It shows you how important the epigenetic control of those genes in the formation of tumor. Next slide. So to give you a sense of how tumor suppressor genes are regulated and function, we use p53, also known as the guardian of the genome, as an example. And there are multiple ways to activate the transcription of p53 genes, such as UV radiation and ionizing radiation, which can cause damage in DNA, and hypoxia, which means the lack of oxygen, as well as oncogene signaling. And the transcription of p53 gene generate p53 proteins. p53 protein is a transcription factor, which in turn can turn on the transcription of multiple other genes that are regulated in multiple other processes, such as cell cycle arrest, DNA repair, apoptosis, also known as post-1 cell death, as well as the blockage of angiogenesis. Angiogenesis is known as the development of new blood vessels. And all these processes are critical in controlling the formation and development of cancers. And there is an experiment carried out to examine the apoptosis function of p53 genes. Scientists use mice cells and expose those mice cells to high dosage of X-ray radiation, and later on they measure the percentage of cells that were still alive after the exposure to the X-ray radiation over time. As you see on the bottom left, the x-axis represent the time elapsed, and the y-axis represent the percentage of cells that were still survival, and which called viability of cells. As you see on the green line, my cell with both functional p53 gene had the lowest viability over time, while the my cell with both non-functional p53 has the highest viability. The reason for that is because when the cell were exposed to X-ray radiation, it caused damage in the DNA. In this case, the DNA damage was too great for the cell to repair. Therefore, they need to undergo the apoptosis to avoid the fixation of this mutated gene. And consequently, the mice cell with both functional p53 has the lowest viability because they can undergo the apoptosis most efficiently to eliminate those mutated genes. Next. And more molecular details on how p53 regulate those process. They lead to cell arrest simply by increasing the expression of the cyclin-dependent kinase inhibitor, which in turn inactivate the cyclin-dependent kinase and cause the cell arrest. And the example here used here is P21, which can inactivate the G1 and S cyclin-dependent kinase and lead to the cell arrest in G1 phase. And at the same time, p53 also activate the transcription of genes that are responsible in DNA repair, such as the pCNA and SP proteins. So there are two possible outcomes. If the DNA can be repaired, they will continue on the cell cycle. However, if the DNA cannot be repaired or if they need too much energy to repair, they need to undergo the apoptosis to eliminate those mutated genes. And p53 regulate the apoptosis while regulating the gene expression of BCL2 and BAS gene. By decreasing the BCL2 gene expression and increasing the BAS gene expressions, they cause the release of cytochrome C from mitochondria. Also, by the way, BCL2 gene is an oncogene which is responsible for helping the cell to escape the apoptosis, and BAS gene are the tumor suppressor gene that activate the cell to undergo apoptosis. And from the release of cytochrome C from mitochondria, it stimulate the intrinsic apoptosis pathway and lead to the cell death of those cells with the mutated gene. Next. And the take-home message here is cancer equal to the excessive growth plus the abnormal spread, which is the metastasis, and tumor arising from a serious event. Therefore, it needs time to develop. It's a disease of old age. And cell cycle is closely regulated by oncogene and tumor suppressor genes, and overactivity of these oncogenes and underactivity of those tumor suppressor genes can lead to the development of tumor and even later cancer. And there are two ways to regulate the gene expressions, epigenetic way and genomic way. And next, and this is the website for our presentations, and that's all. Any question?
So what is the difference between in terms of what the how they prevent or how they cause cancer? Yeah, the proto-oncogenes. Okay, so the proto-oncogenes, they what they do is they regulate cell division. So once it becomes an oncogene, it keeps going, keeps dividing. So how the proto-oncogene regulates it, it just makes sure it doesn't keep on dividing, it just regulates the proliferation of the cell. And then the tumor suppressor gene, what it does is that once it goes to the part where the tumor suppressor gene works, it regulates it. It checks on the gene and checks if there's any mutations. And if there is a mutation, it will stop the cell cycle. where it's at, and it'll try to repair the DNA. So so the way that when it doesn't work, it would just everyone won't stop it. It won't even check it. So it could just be mutated and it'll keep on going. The cell cycle will continue, and that means that the proto-oncogene would keep on making mutated gene or will work with the mutated genes and just keep on making bad cells and replicating it. If OncoChip keeps going, oh, sorry, if you want to answer. Shin.
Yeah, so to answer the question is the oncogene, if the activation of oncogene, they can lead to the development of tumor. And the activation of tumor suppressor gene, they can add to suppress the tumor development. So they both involved in the tumor development, but one, the oncogene is add to lead to the tumor, but the tumor suppressor is actually suppress the tumor. So that's the difference.
Yeah.
Yeah, the epigenetic basically mean they can cause the activation of the oncogene, which lead to cancer, as well as they can silence the tumor suppressor gene, which also lead to cancer. But they does not need to be the direct alteration of DNA sequence to cause this activation and deactivation. There are also other way that, such as modifying the structure of chromosome or add some like methyl group or acetyl group to the DNA, which can also add to silence or activate those genes. Yes.
Yeah.
Here. Sorry. Just a little question. For the overactivity of the oncogene, is that just considered like a mutation, or is it just like, overexcited? Could you further explain that?
Yeah. So basically, when a proto-oncogene becomes an oncogene, it means that it's overactivated, so it just keeps going. But if it was a proto-oncogene regularly, it would just it'll regulate cell division. So that's why when I said the overactivity of proto-oncogene is basically of oncogene is basically just a proto-oncogene becoming an oncogene where cell division just does not stop. It's continuous. It is a mutation in the proto-oncogene that converts proto to onco.
Okay, thank you.
Mm-hmm.
Yeah. Can you go back to the slide with the epigenetic alterations? Do you guys know like the mechanism behind that, like how it happens, or do we just know that it happens?
Actually, I don't quite know what is the mechanism. You just know that sometimes the DNA, if it's methylated, it actually can condense those DNA like less accessible to those like transcription factor and RNA polymerase. But in terms of what like mechanism the methylation caused the silencing of those tumor suppressor gene, I think just those scientists they happen to discover that in those people genetic sequence of those people with cancers, and they examine their genome sequence. They happen to see that in those sequence, they happen that the promoter region often are methylated, so they have discovered the chain of like the methylation maybe cause the silencing of the gene. But in terms of mechanism, I might leave this question to the TA.
Okay. Yeah, it does. Thank you.
Yeah, for metastasis, does it only occur like via blood? Does it only transport through blood?
So I believe it does because from what I know, metastasis is when the cancer is able to travel to a foreign tissue. So in order to get there, I think it has to enter into some sort of capillary or vessel.
Also, like to add on the question, the reason that they're able to undergo the metastasis because, besides the mutation on oncogene and tumor suppressor gene, they also mutate other gene that enable the cell to actually destroy those, like the CAN, those destroy those connection between cells and also destroy the kinetic tissue in the basal lamina as well as the underlying kinetic tissue to enable them to actually travel to the blood vessel, then travel to the secondary tumor sites. So just to keep in mind that they need a certain gene to be mutated to actually travel to the capillary.
Okay, thanks.
Right. Um, isn't it because it accumulates more and more mutations every time it proliferates?
Uh, okay, my understanding is maybe as the cell they mutated, the cancer cell keep going, and then more and more proliferation and more cell there. So when they to the point that there are no enough oxygen for them or not enough nutrient for them, so they need to find a new place to migrate so they can continue to grow.
So, like just to add to, I think also because like if you're to see like in this example right here, you kind of see that this cell got a mutation, and then that same cell was able to make clones of itself, and then it was able to acquire another mutation, but yet it was still able to make more clones of itself. So eventually, it has like a stock of cells that are mutated already. And so now these cells are able to divide faster and undergo more accumulations of mutations. And then from there, eventually acquire like some sort of trait that allows them to like break through the lamina and progress. Is that what your question's asking? Yes. Why does it make it more fit? Like, are you talking like in regards to like um well, because it's when it divides faster once it accumulates more and more mutations because the right.
I've
Because each time the cell divides, it's it's accumulating more mutations, and these mutations, yeah, but also at the same time, the more mutations a cell gets, the it's going to
can I answer the question?
bypass more.
No, actually, maybe actually it kind of like a selective mechanism. Like those cells, they can escape the apoptosis, and those like normal cells, they actually undergo like 50 division, they already die. But those cells, they actually are selective. They kind of like can keep continuing to grow. So at the end, only the cells that are able to grow like continually, they kind of let over. And then if the cell continue to accumulate at the point that they don't have enough oxygen and nutrient, they need to find a new place to grow. And the reason why they become more and more like progression is because they cannot be like killed like other normal cells. And they see that, oh, there's advantages, they continue to mutate it. And those at the end, only the one that's selected over are those mutated cells are cancerous, so they can survive. Mm-hmm. 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 Yes. Mm-hmm. Yes.
So it's kind of like natural selection kind of act. Yeah, so just to clarify, it's because natural selection kind of acts on like different subpopulations of cancer cells that kind of pressure them into picking up like mm, okay.
Yeah, you don't have to pronounce my first name. It's fine.
Yes.
Okay, hi, we're group 6, and today we're going to be presenting on the mutational experiments that were utilized in analyzing the cell cycle.
So from last week, we had a presentation on cell cycle, and just to reiterate, it is an orderly and tightly regulated process to carry out cell division. There are 6 phases, which we all know: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Yeah. Okay, well, the main points of this presentation, we will be going over the search up for master
Oh shoot!
regulators.
Oh my god! My thing wasn't on. I'm so sorry. I thought I
It's
was
all good.
talking. I was talking and like okay. The main sorry. The main points we're pretty much going to talk about is like the search for master regulators for the cell cycle and the mutational experiments, which there are 3 of. One being the cell fusion experiments, which we're also going to discuss molecular bases of cell fusion. Foci experiments, which include the MPF purification and maturation assay, and temperature sensitive experiments, and including the cycling discovery. Sorry, I thought my thing was already open. Yeah.
Okay, so before going into the cell fusion experiment itself, I'll be talking about the molecular basis of cell fusion. So we all know that DNA replication begins at the origin of replication, which can be identified as the origin recognition complex, or ORC, which is shown right here. In the G1 phase, ORC recruits CDC6 and CDT1. Then it also recruits MCM, which is a DNA helicase, and it assembles on the flanks to create this pre-replicative complex, also known as pre-RC. Next, SCDK triggers S phase and then recruits pre-initiation complex while phosphorylating ORC. During this step, CDC6 is phosphorylated, which becomes degraded, and there's also the inhibition of CDT1 by geminin. Now the MCM helicase is activated to unwind DNA. This initiates replication. After this, there is an mCDK activation, which initiates chromosome segregation into 2 daughter cells. So even if there is SCDK in the G2 and M phase, DNA replication won't occur because there needs to be all the other proteins present, such as the CDC6, CDT1, and MCM helicase for the organization of the pre-RC, which is only in the G1 phase. So this means that the cell has to go through mitosis in order to start again at the G1 phase to recruit the necessary proteins for replication. Now applying the molecular basis of cell fusion and moving on to the actual experiment. These fusion experiments took place in the 1970s. The experiment is basically just isolating and fusing cells of different phases to form hybrids. So here in the first scenario, we see that we fuse S cells with G1 cells, and as a result, the G1 nucleus continues into S phase, which is where DNA replication takes place. Now, if you fuse S cells with a G2 cell, we can see that G2 stays the same. This experiment suggested the presence of a S phase promoting factor, since this promoting factor initiated DNA replication in the first scenario. However, in the second scenario, the G2 nucleus failed to initiate DNA synthesis. So there's something within the G2 cell that is preventing DNA replication until after mitosis has taken place. Now, if in this third scenario, if you fuse G1 and G2 cells together, G1 cell enters S phase based on its own timeline, while G2 nucleus stays in the G2 phase. So this just shows that G1 and G2 are independent of each other.
When it comes to the maturation assay, maturation can be induced by injecting the nucleic contents of the maturing oocytes into another oocyte that hasn't been treated with the hormone. Pretty much, Masai and his colleagues used the maturation assay and show that MPF displays the properties of a mitotic inducer. You can go to the next one. Okay. An oocyte is basically an immature egg that will mature and become an ovum through the process of ovulation. In the early 1970s, Masai was working with frogs in which fully grown oocytes are naturally arrested in the G phase of the cell cycle. In his experiment, he found out that MPF activity is higher in the cytoplasm of those going through oocyte maturation than those during the early cleavage divisions of a fertilized egg. So what he did is that he took the cytoplasm from a unfertilized egg that was stopped in metaphase and microinjected that into an immature oocyte that is in the G phase. And the picture does show that as a result, he observed that the microinjection was able to trigger the transition from G2 phase to M phase, which the diagram also indicates right there. This experiment demonstrates that the unfertilized egg stopped in metaphase contains a maturation promoting factor, also known as MPF, that drives immature oocytes into G phase into mitosis phase, also known as the M phase. You can go on to the okay, cool. The purification of in 1988, Manfred and James purified MPF, but the purification of MPF was difficult because of not knowing their identities. There are millions of proteins in the cytoplasm, so to inject the cytoplasm into the oocyte, which allows the cells to enter into mitosis, would make it difficult to identify the exact set of proteins. Ultimately, the MPF was purified through a chromatographic experiment, which allowed the use of sequential steps to purify the proteins. They identified that MPF consists of two proteins, one being P32 and the other being P45. P32 was also discovered to have protein kinase activities, which means it phosphorylates other proteins. Then, yeah.
So next, I'll be talking about the cyclin discovery. Cyclin was discovered by Tim Hunt in the early 1980s when he was studying protein synthesis during synchronous cell cycles in sea urchin embryos. By definition, cyclin is a family of proteins that drives the events of the cell cycle by activating dependent kinase enzymes, which we know as CDK enzymes. These enzymes are necessary for the synthesis of the cell cycle. For the experiment itself, Tim Hunt had to analyze newly synthesized proteins in the cell by using a pulse chase technique. So first, he took the sea urchin embryos and pulsed them with 35S methionine. Proteins were then isolated and extracted and were subjected to SDS page and audio radiography. As a result, Hunt observed an oscillating pattern in which proteins appearing at one stage of the cell cycle and disappearing in another stage. So as we've seen in this image, we can see that cyclin are both present during mitosis. And in fact, Hunt observed that cyclin A and B appeared in the M phase and disappears during interphase, which can also be seen in these darker lines. This suggests that the cyclin is being continuously produced and being destroyed by proteolysis. In lecture, we also learned that the cell cycle control system is based on these CDKs, and these CDKs are successful when they associate successfully with different cyclins to trigger different cell cycle events. Later on, they'll come to find that what Masui identified in biochemical analysis P45 is actually cyclin B, which Tim Hunt identified while working with sea urchin embryos.
Next, we'll be talking about temperature-sensitive mutation experiment. This was performed by Lee Hartwell and Paul Nurse. They used temperature-sensitive yeast mutants for various reasons. First, their genomes are really small. They're haploids, so it's easy to isolate the mutants since only one copy of the gene is affected. Gene manipulations are straightforward. You could do cloning by complementation. It's easy to pinpoint prominent cell cycle control points. So later on, we're going to see that both the temperature sensitive mutation experiment and the oocyte experiment actually led to the identification of the same specific proteins in cell cycle transitions, but just through different approaches. This temperature sensitive mutation experiment utilizes a genetic analysis, while the oocyte experiment was using a biochemical approach. And so essentially they chose to analyze yeast because it's easy to, again, dissect the cell cycle in yeast, and because they have very simple choices to make. So for example, if you provide food, yeast cells are going to keep dividing. And if you starve them, they're going to enter into sporulation. Secondly, it's easy to determine based on cytological analysis. If you simply look at the size of the bud and bud formation process, it'd be easy to determine what stage of the cell cycle it's in. And also, it's important that a mutant can't complete, if a mutant can't complete a cell cycle, it's not going to propagate. So a CDC mutant can only be selected and maintained only if the phenotype is conditional. So that being said, most conditional cell cycle mutants are temperature sensitive mutants. Therefore, you can shift between low and high temperatures, and that can be used to study the transitions and maintain the mutations to do genetic and biochemical analysis. So if you look at the right, there is a diagram. It's showing the budding yeast S. cerevisiae and the fission yeast S. pombe. So basically, this shows that cell cycle progression in the budding yeast was basically the bud is being formed during G1. The cell cycle commits to cell division, the bud grows, and then the bud separates during the different cell cycles. You can see an arrow pointing, it says the size control point for the budding yeast is between G1 and S, and this is where the cell is going to commit and determine if it's going to make a bud or not. At this point, the cell is checking if there's a favorable environment, if and that just means if there's adequate nutrients, if it's the adequate size, if it has the appropriate growth factors present in order to commit and enter the cell cycle. And if the environment is not favorable, the cell's going to wait for a favorable environment or enter into G0 phase, which is just a resting state. If you look at the bottom, you'll see the S. pombe fission yeast. And here, the cell is again checking for nutrient availability, but also if the DNA was completely replicated, if any DNA was damaged. If the DNA is okay, the cell is going to proceed with mitosis, and if not, the cell will wait for repairs. Here, on the second diagram, this is basically a summary of their findings. So as aforementioned, these are temperature sensitive mutants. So on the first column, you see budding yeast CDC28 mutant. So the CDC28 is a master regulator of cell division in budding yeast. So they identified that a CDC28 mutation at permissive temperature is going to make a proper bud and undergo proper cell division. But as you can see, in restrictive temperature, it's going to fail to divide and make a bud. And that means that the arrest point was between the commitment cycle, meaning that the G1 to S transition is arrested in this mutation. In the second column, you'll see CDC2, 13, and 25. You'll see that these mutants failed to divide. They grew properly, but they failed to divide after achieving a particular size. meaning that the restriction division point that happens is between G2 to M, which means that these cells have a problem in making a G2 to M transition. In the last row, we see that they identified one more mutant, which has a completely opposite phenotype. It's the WE1 mutant. Instead of making regular size cells, these are going to make extremely small cells, which suggests that there is an unrestricted entry of these cells from G2 to M transition. So this is indicative of an unrestrained cell division. And later on, we're going to see that in retrospect, where these three experiments that we've been talking about, ultimately these mutants, the CDC28, CDC2, CDC13, and 25, these identified only two proteins, which are going to end up being the cyclins and CDKs. Here, so once you identify the mutations, it's going to be easy to clone genes in yeast. The general strategy to clone genes is you take a mutation and you transform the mutation in a library of yeast DNA fragments, which represents all possible genes in yeast. Then you look at which clone rescued the particular mutant. That suggests that that particular gene is responsible for creating the particular mutant phenotype. Then you can go back and sequence the gene and identify the protein product, which was responsible for making this transition. And in this slide, we're basically seeing all of our experiments merging, our biochemical and genetic analysis coming together. It turns out that these proteins in the aforementioned experiments are related to one another. So it was found that CDC28, which we said was in budding yeast, and CDC2, which we said was in fission yeast, encode related analogous protein kinases. It was also found that Masoud identified the P32 subunit of MPF. Well, that P32 subunit ends up being the exact same as CDC28. In addition, genes encoding CDC2 mutant rescue CDC28 mutant. Also, genes encoding P32 rescue CDC28 and CDC2 mutants. Therefore, it's concluded that P32, CDC28, and CDC2 are all the same proteins with the same function. They just encode CDKs, which are cyclin-dependent kinases. This also gives insight as to a similar kind of gene that they all contain. They will all consist of two subunits, which one has kinase activity, which I said was the CDK, and the other is going to be a cyclin. Monsieur identified the P45 subunit, and that actually ended up being the same as cyclin B that Tim Hunt also identified. Also, CDC13 turned out to be a cyclin, and they later went to show that cyclins bind to CDKs.
So what's important in all of this is that cyclin-dependent kinases are present, whereas cyclins itself are produced only during the cell cycle transition, which we saw during the Tim Hunt experiment. This means that cyclins are cycling through mitosis and bind to cyclin-dependent kinases, therefore activating this CDK and cyclin complex, which then triggers the events to the different cell cycle events. Thank you. Oh, yeah. this experiment just suggested that there is an S-phase promoting factor, since in the first scenario, we fused the S cell with the G1 cell, and the G1 cells was driven to the S phase, which means that there's something that triggered, that there's an S promoting factor just to trigger it.
Also, I'll add to that. It also suggested the presence of a mitosis promoting factor in mitotic cells.
Could you talk about or go over again why the temperature sensitive mutants or organisms were important to the experiment?
I'm sorry, could you repeat the question one more time?
Yeah, could you go over why using temperature sensitive mutants were important to the experiment?
Yeah, so basically, you don't want to hinder kind of like the mutation process. So there are temperature sensitive mutations, and so like when it's at a permissive low temperature, you're going to allow a mutation to pass through, and in a restrictive temperature, it's going to hold the cell cycle like at a specific state, and that's what allows for the analysis of just the different master regulators of the cell cycle. Does that answer your question?
Yes, thank you.
What would occur if a CDC mutant was allowed to propagate, and how would it affect, in the case of the temperature sensitive mutation experiment, how would it affect the yeast cell?
So sorry, one more time.
In the case of the temperature sensitive mutation experiment, what would occur if a CDC mutation or a mutant was allowed to propagate, and how would it affect the cell?
I'm not sure. I'm not sure they went over this in lecture or if I understood that.
I'm not sure what your question is asking, because basically this experiment is saying these mutants, like for CDC28, if it has a mutant, then it fails to bud. And in the CDC2, 13, and 25, it fails to divide. While there's a W1 mutant, it fails to grow.
Okay, yeah, that I guess that's what I was asking for. Yeah, cool. Thanks.
Yeah.
I guess I have a question, or maybe more of a clarification. On this slide, actually, the last mutant, the W1 mutant, how does unrestrained, oh, never mind, I see it. It's so the it's not able to fully develop, so that's why it's short.
Yeah.
Short. Okay.
Yeah, so the WEE1 is it's a kinase enzyme that phosphorylates the cyclin CDK complex in the inhibitory site, and that inactivates the protein, and it adds an inhibitory phosphate. And when you have a mutant in that, you can't put any more inhibitory and phosphorylation, and now the cells enter into mitosis in an unrestrained way.
So like they would stop early in cell division, or like they would stop maybe like before the G2 phase?
Yeah.
Okay. Thank you.